Today we will talk about RNA-seq data after it has been processed. So how do we analyze it and how do we explore what is in our table of expression? So we've so far covered the first three or the first two sessions is what we covered, and we'll cover today the third one, which is how the data is generated. So what are some library preparation steps? What are some of the initial steps of the analysis? Basically, how do we start processing the data so that it is structured so we can do the analysis? And what does it essentially represent? So what are we quantifying? And importantly, some questions about data preparation techniques were asked in the last session. So Harpreet is adding a few videos that I think are going to be very useful for those who are familiar with the wet lab techniques of how the data is, how the samples are prepared and what is being extracted and what is being put in the sequencer. So please take a look at those videos. And also there's a publication that you can read through if you want the technical details. You'll find those in that publication reviewed from a research perspective. So about the progress, we've been seeing that many of you have started taking the online courses. And of course, you probably found that the same questions that are being posted in the sessions are being asked in those quizzes. That's just to help you remember how some of the terms fit into what we are discussing. And also some of you have been taking some of the coding assignments. Remember that the coding assignments are designed to supplement the sessions that we have here or that are being recorded if you're watching the recorded versions. And a lot of times they go from very basic and very simple to a little bit more and more advanced, offering you some flexibility. So in many cases, what we will do in the sessions is going to start you off thinking about what exactly are we trying to do. For example, we can open a table of expression in Excel or OpenOffice or some kind of a table manipulation software. Well, you also can open that table in R by loading that table and viewing what's inside. If we do an analysis like t-test, we can perform the same functions in Excel, we can do it in R, we can do it in Python. So what the program will help you understand is what is the thinking behind many of these different approaches. What do they provide us with? So what's the output of that analysis? But how to do them, you are free to decide whether you want to just rely on tools that are visual and interactive, or do you want to also learn some of the coding? So it's great to see that many of you have an interest in those coding tutorials, and we are hoping for your feedback as well. We're going to be reviewing all of the feedback after the program is over, making some additional changes to those. So please return to those courses, and just to kind of point out where to find those. So you can go both on the coding site, which is code.omicslogic.com, or the same website is learn.omicslogic.com. This is something that we are just kind of finalizing right now. But here under courses, you will find you should have access to all of these courses. So you have a few different ones on just dealing with basic sequences. And you also will find some specific to transcriptomics right here under this course. So please take a look at those as we go through this session. Okay, let me just check chat real quick. Okay, thank you, Beepsa. Yes. Okay, so going back to our sessions, I did want to highlight a few profiles because we started off with just kind of setting everything up. So there's a lot of progress that you can see on our program page, and some of the people that have joined have completed their profile. So it's good to know who else is in the program. So Francisco Ferreira, for example, Dalia Mihaisi, who is a PhD student from the UK, and Lindsay Rhodes also completed her profile. She's an associate professor at the Florida Gulf Coast University. So if you have not completed your profile, we just ask that you do complete your profile. Again, once we get to the stage of working on projects, you'll be able to interact with other participants and get to know them and maybe develop a project together. It's always helpful to get to know other people. And so to help us with that, if you can add some information about you or have some even description of another profile that you have on social media so that people can connect with you, I think that could be helpful. So let's review what we talked about in our last session. We discussed how the abundance of mRNA in a sample, in a bulk RNA-seq sample, is the objective of this analysis at the first step. When we want to prepare a table of expression, we want to compare the abundance of mRNA per sample, and there are many different techniques of doing this. The advantage of next generation sequencing is that we get the exact sequence of short reads. And so when we look at the sequence of those reads, even though they are short, by mapping them together onto reference genome, we can identify many different things from a single analysis. So we can look at the sequence of reads and map them onto known genes, or we can look at alternative splicing, which also comes from the GTF file where many different transcripts are lined out. And we can also detect some novel combinations of exons by doing a more in-depth analysis and identifying junctions between those short reads. The quality of the reads and the length of the reads determines how accurate our analysis can be and how trustworthy the different details that we find. For example, if we're talking about novel transcripts or even variations in exons, if we have longer reads and higher quality and greater abundance of reads, we will have a more reliable analysis as a result of that. So those steps matter. And one of the ways that we can make sure that the initial raw data is high quality is by going through some pre-processing steps. And so it's important to understand the logic behind these different pipelines as we look at the data preparation, how it was prepared, and we think of the objective. Where do we want the analysis to go? So when we come to this pipeline builder, and the demo pipelines are there to help you try out many different combinations of these tools, the logic of this pipeline builder is that we have some basic steps like pre-processing, mapping on the genome, construction of isoforms, and mapping on transcripts. And what we can do is we can determine whether, based on what we know about library preparation steps, whether we need to do pre-processing or not, whether we want to map on the genome and identify novel junctions and construct novel isoforms, or just map on transcripts. Now, there are also additional types of methods like Sailfish, which do not require the mapping. They just look at a database by looking at k-mer matches from the database of known transcripts. And it's a much faster method, but it loses the detail of knowing alternative splicing events and being able to determine novel isoforms. However, in terms of knowing what the meaning of those novel transcripts might be, of course, will require some additional interpretation. So the basic steps, as we reviewed last time, are pre-processing, which is working with the individual reads before we know where they align, making sure that the reads that we have are cleaned from any additional sequences, and reads that are unexpectedly abundant are typically eliminated or moved down to the level of transcription that is expected from the overall file that we have. Then mapping onto the reference genome or the reference transcriptome, and then finally quantification, which could happen in a number of different formats. And in Transcriptomics 1, you will go through all of these steps and find a few questions to make sure that you understood the content. So let's go through some of these questions. So quiz question number 1, if you already completed in Transcriptomics 1, you have done this. Which of the following statements regarding transcription is correct? 1, the central dogma of molecular biology describes the two-step process, transcription and translation, by which the information in genes flows into proteins, DNA, RNA, protein. Transcription is the synthesis of an RNA copy of a segment of DNA. RNA is synthesized by the enzyme DNA polymerase. So as you read through these answers, please put the number that you think is correct in the chat box. Number 2, the central dogma of molecular biology describes the three-step process, replication, transcription, and translation, by which the information in genes flows into proteins, DNA, RNA, protein. Transcription is the synthesis of an RNA copy of a segment of DNA. RNA is synthesized by the enzyme RNA polymerase. 3, the sum total of all messenger RNA molecules expressed from the gene of an organism are produced during transcription, with exactly none of these RNA molecules being produced during translation. And 4, more than one answer choice is correct. Okay, so NG is saying 2. MHC18 is also saying 2. If you could have your full name, so it will be easier to know. Okay, does anyone, okay, Rekunli is also saying 2, right? And Doug's computer is saying 4. Okay, so the correct answer, Dahlia, okay, thank you. Dahlia is also saying 4. All right, so yes, the correct answer is 4. If you read through, we know for sure that number 1 is not correct because you can see their DNA polymerase. So it's obviously not DNA polymerase, but RNA polymerase. And we know that number 3 is also correct because the sum total of all messenger RNA molecules expressed from the gene of an organism are produced during transcription, with exactly none of these RNA molecules being produced during translation. What is produced during translation? It's the protein. So let's go to question number 3. Which of the following statements in regards to the central dogma of molecular biology is true? 1, an exon is a long stretch of non-coding DNA found between introns or coding regions in a gene. Genes that contain exons are known as discontinuous or split genes, as the coding regions are not continuous. Exons are found only in eukaryotic organisms. 2, alternative splicing is a process by which exons or portions of exons or non-coding regions within a pre-mRNA transcript are differentially joined or skipped, resulting in multiple protein isoforms being encoded by a single gene. 3, gene isoforms are various RNAs that can be produced from a single gene. Isoforms are also known as splice variants, referring to the process of splicing or cutting out introns and sometimes skipping exons, thus getting different mRNAs from the same gene sequence using different combinations of introns and exons. And 4, more than one answer choice is correct. Okay, so let's put a separator between the previous question and this one. So please, again, put your answers and we'll have a little discussion about this. So as you think about your answer, probably most of you have heard about DeepMind, the prediction of proteins from sequence. And here, I think this has to do with that capability. So one of the exciting reasons why that is a breakthrough in biology is because about 150 million different proteins exist out there. What we know from structural biology studies like cryo-EM microscopy is that there is about 150,000 known protein structures. So what we don't know are how many different proteins that are produced as a result of variations that we have not had the chance to study using microscopy. So there's a huge variety of proteins out there, and the way that this huge variety could be encoded efficiently in the DNA is by using the system of exons and introns and alternative splicing. So this mechanism is important. Okay, so many of you have answered number 3. Some of you, or many of you, also have answered number 4. So let's see, why is number 1 incorrect? So an exon is obviously the coding region and the intron is the non-coding region. Okay, so number one is not true. Number two, alternative splicing is a process by which exons or portions of exons or non-coding regions within a pre-mRNA transcript are differentially joined or skipped, resulting in multiple protein isoforms being encoded by a single gene. And number three is gene isoforms are various RNAs that can be produced from a single gene. Isoforms are also known as splice variants, referring, okay, so both number two and number three are correct. So number four is the right answer. So what happens in alternative splicing is that exons could be joined together or skipped, and then we have the result of that is what's called a transcript or a protein isoform. So it's a variation of the same protein. So number four, again, is correct. Okay, so let's review RNA-seq briefly. The reason behind this great variety of proteins that are being produced from a single gene is why we need to sometimes look at mapping those reads onto the reference genome. And when we understand how the different exons are spliced together, we are really measuring isoform or transcript expression. So isoform is an isoform of a protein. Transcript is a transcript of a gene that is being transcribed. And so when we measure the level of mRNA expression, we are looking at those levels. What is an expression of a gene? An expression of a gene is simply all the expressions of that gene transcripts added together. So in some sense, it's an inaccurate measurement, but it's an easy measurement for downstream analysis, because the more variation you have at this quantification level, the more variation you're going to find as you start trying to piece this puzzle together to interpret the biological background of whatever you are studying in your samples. And so many times, and what we will do in the next several sessions, is assume that there is no transcripts. Transcripts are just a combination of all of the, so genes are a combination of all the transcripts that we have in our data set. Now, as you go through the different sections, you will find some information about each one of these pre-processing steps. And we will also add some additional mention of the specific details for each one of these methods. Oftentimes, you can just click on that and get a pop-up, and that pop-up will explain exactly what this method does. So please try and work through the demo pipelines and also perform the assignments that are built to illustrate how some of these techniques vary. Now, a simple way to test whether you are familiar with the different names and the different meanings of these algorithms is to try and run some comparisons. So you can actually take a look at the same data set being processed with multiple methods. So as we go into the project and you identify a data set, you might want to explore how variation in the different algorithms plays a role in how this data could be quantified and measured, ultimately producing a table of expression. So this level quantification of levels of expression is a very important step. After that data set is produced, we can start exploring or working with that data set. And this session will go into how you can do that, and especially what are some of the important considerations as we pick methods for statistical analysis, because these methods of statistical analysis have some assumptions built into them. So they have to do with what the data looks like at the level of statistics, like distribution, minimum, maximum, etc. So these are some statistical properties that we are trying to compare. So I will pass it on now to Dr. Harpreet Kaur, and she will guide you through how to take this data from the resulting pipeline and then be able to do downstream analysis.
Thank you, Ilya. So let me share my screen. So hello everyone. In the last session, we talk about how we can generate a well-structured gene expression data table from the raw sequencing reads. So today we are going to talk about how we can perform various kind of statistical analysis and how we can explore that gene expression table to scrutinize any biological significance from the data. So in the last session, we talk about how we can perform log scale transformation in the Excel. So today, first of all, we are going to perform quantile normalization on that data set that we have obtained. Last time we obtained a gene expression table after running the demo pipeline that contains FPKM value. So first of all, we are going to perform log scale transformation and quantile normalization on that data. So let me share this file that I have already posted in the forum. So let me share that file that we have obtained after running the pipeline last time. So I'm sharing the link of that file. So you just download this file. So this is the data set that we have obtained after running the pipeline last time, which contain FPKM values. So once you download this file, just type yes in the chat box so that we can proceed to perform normalization on the data. That's great. So now if you log into the server TBinfo, okay, so I'm posting the link. So this is the link for the server where we are going to perform quantile normalization and log scale transformation. So once you're able to log in, so on this interface, here we will find various sections for different kind of analysis. So under the data mining, so under the data integration, and here we have a section that is data mining. Here we have the third option that is utility. If you click on this option, so how many of you able to log into the server and able to find the utility sections? That's great, David. Okay, so just click on the utility. So then we have to upload that txt file, the gene expression table that we have obtained after running the RNA-seq pipeline. So if you go to the file upload, here under the first option where we are going to upload our file, so at the file, so you have to click on only the utility section first. David, you just click on utility options. Then you will direct it to this page where you need to upload files. So then we will continue. So here after we have already uploaded file. Okay, so you are clicking on actually drop down. You need not to click on drop down. Okay, I can go again. So let me just show you. Under the data mining, we have third option. You need not to click on drop down, you just need to click on utilities. So those are the demo. If you click on the drop down, those are the demo pipelines. So this pipeline don't take much time, so we are directly running this pipeline. So once you click on that, then you will get directed to this interface. Here you will get this option, that is file upload. If you click on file upload, here under the first option, that is add files or upload files, you just need to click and need to upload that particular file. And remember, your file should be always in a TXT format. And make sure that your this file should not have any blank line at end of the file. So once you're able to upload this file, then go to the continue. Then we can click on start. David, is it working for you? Okay, that's great. So once you click on that, then under this first option, we are going to perform quantile normalization. So if you click on this quantile normalization, here you will get you have to set the parameters. So the first parameter, what it is asking, that transform table to LN, that is log scale transformation, whether you want to perform convert this table into the log scale transformation. So if we click on yes, then we have to select the threshold. So as once our last time we have looked at the data, so once we able to perform the log scale transformation, our data variance values start varying from, that is 0 to maximum 20. So here we are selecting a 5 threshold, that we want to select this particular threshold that will filter out our cutoff only for the genes. So once we able to select the parameters, first is we are performing log scale transformation, then we are selecting the threshold to perform quantile normalization. So then you just save it. So the next option is, it is asking that whether you want to perform PCA. Here we are skipping this step today, as we are not right now not interested to perform with data visualization. So if we want to perform data visualization, we can select that. So right now I'm skipping that. So here our aim is to perform only log scale transformation and quantile normalization. So then we will, with the end, we are going to complete this pipeline. The next, it will ask, you want to name this pipeline. So what we are going to do, QN pipeline. So once you named it, next you need to click on run pipeline. So it will take 2-3 minutes to complete this pipeline. So now where you can find whether your pipeline get run or not, next just click on my pipelines. So right now it is just showing the progress, only the 0%. So it will take a few minutes. So how many of you are able to run or complete this pipeline? Add it to it. Okay, that's great. So you can refresh it. So it will take 4-5 minutes. So you need to check whether your pipeline get completed or not by refreshing. So what we can do right now, first of all, here we need to paste this pipeline gene expression table just to compare. So this was our original file, original data. So I'm going to paste it in Excel. Okay, so yeah, it will take 2-3 minutes to complete it. So let me look at my pipeline, whether it get completed or not. So I can refresh it. Now my pipeline already get completed 100%. It shows the progress done 100%. So if I click on this pipeline, now under the download pipeline, I need to click on this folder. And here we will get this output. So this is showing given pipeline, this particular file, threshold 5 normalization, normalized text. So this would be the quantile normalization value. So now I can check whether what is the difference after the quantile normalization. So this was my normalized data. So you can refresh it as well to check whether your pipeline get completed or not. So this is quantile normalization. This is FPKM values. So what we can do, so here I'm taking this particular to compare what is the effect of quantile normalization. I'm taking this second chain. This is with the raw values, and I'm taking gene ID. So so how many of you able to complete your pipeline and able to get this output? So just need to, okay, that's great. So now let's make a simple bar plot to compare after what is the effect after the normalization. Here I'm taking only a one gene and then I'm comparing its expression among different samples. So this gene, when it has FPKM values, and now I'm considering so if you look at this, if you look at the y-axis, here our values in case of when we have this blue color plot is representing when we have FPKM values. So if you look at this plot, first of all, just look at y-axis. It represents here variance in the expression range is from 0 to 30, nearly 25,000. Now, if you look at this after the quantile normalization, now the range of values is from 0 to 10. So this is what the first it reduced that large wide range of variance. If you look at this, it looks like here in the ER positive sample, this gene is highly expressed, while in triple negative it looks like this gene is low expressed. But when after the normalization, if we looking at this, here we don't find this difference. So this difference get disappear. So that's why it indicates normalization of a data is vital to derive any biological interpretation from your data. So how many of you able to generate this bar plot? That's great, David. So any one of you able to produce bar plot with your data before the normalization and after the normalization. Had you tried? So that's great. Francisco. So this shows why normalization of RNA-seq and RNA expression data is vital to derive any biological interpretation, especially correct biological interpretation. Okay, so this is how we can perform log scale transformation and quantile normalization using T-Banford server. Once you have data, that is have FPKM value, have a gene expression table, and then you can compare that. So the next, how we can upload this data and visualize our data and perform log scale transformation in the R or the Python. So today we are going to demonstrate to you how we can perform, first how we can load data in R, then we can do various types of manipulations on our data, and then how we can visualize our data using box plot, bar plot, or scatter plot. So now let's go over the another platform, that is called OmicsLogic. Let me paste its link here. So Jefferson, advantage of quantile normalization. It's usually normalization technique is not always works or fit with every data, but for the if you are especially comparing or taking a data set from different platforms or from different studies, usually in literature it has been shown that quantile normalization is one of the best method. Although it's not it's not a 100% useful for every single data, but most of studies quantile normalization shows a better results, and especially if you developing a particular prediction model, machine learning. So in that case, if you taking a data set from different types of studies and or different platforms, in that case quantile normalization shows a better advantage. Although you can perform G-score normalization as well, but it's not like a particular normalization is always suggested for every kind of data set. So you can always perform multiple types of normalization. So if you go to the courses, so let me just share this link with you. So just let me know how many of you able to log into this OmicsLogic platform. So here under the different courses like Introduction to Data Science, as well as under the Transcriptomics, we have these courses where we can learn how we can visualize and do various kind of manipulations on our data. So just go to the load more. So just go under data visualization in R. So how many of you are there on this platform? Okay, that's great. Okay, so here whenever we are giving on any lesson, so first section is always about understand the problem, where exact the logics and the problem is explained. So while clicking on complete, then go to the next. So in the next section, usually learn the code. Here the code blocks are each code block explain exactly what's happening in the code, and each step is explained in a stepwise manner. Once you're able to complete it and understand it, then you go to the next. So the next is here how you can load a data. So I have shown you this particular link. Let me copy the same link that let me take the same file that we had. So here you need to do, so I'm just, I just need to copy the link of the file. So first of all, in R, how we can load a data. So here, as R is object-oriented language, so we always create an object where we store our file, our data. So here with the read.table, that is this command is used to read any file, especially if it is txt file, or if you have CSV file. In that case, you even write read.csv. So here I'm using read.table. Then we have to can you paste the link to your quantile normalized data? Yes, we can use that as well. Here I'm just going to perform first log scale transformation on our data, and how we first we are going to learn how we can read of any data in our R, then how we can do various kind of manipulations. So here I'm taking the same file that we have used, that contain FPKM value that we have used to perform quantile normalization. So with the read.table command, we can read our file. Then we need to provide the link of this file. Then we have to define whether this separator, that is how the columns in this file are separated from each other. So here in this file, they are separated with the tab. Then we have to define whether header is present in the file or not. So let me just maximize it. Header equal to true. Then we can also define row names, that is row dot names equal to 1. So I am also copying this particular command in this chat box. So now to check whether this file get loaded or not, what we can do, either we can head this file or we can check the dimensions of the file. So how we can check the so with the dim command, that is dimension, that is there. So we can check the dimension of our file. So if you look at this, here we have 3310, that is 3310 rows and 20 columns in this file. So with simple dim command, that is to check the dimension of a file. So once we able to load our file, next we can find the summary statistics. Summary statistics, what are the summary statistics? So last time, if you remember, with which plot we can check summary statistics? Anybody remember which plot represent the summary statistics of a data? Yes, box plot. So in R, apart from the box plot, a summary. The simple summary command will provide you the summary statistics of a data. So if you look at this now, here you will find the details like what is the first quartile, what is the minimum value, what is the maximum value. So then what is the first quartile or the third quartile. So if you look at this data, the minimum value is 0 and maximum value is something like this is 9, 1 and 9 to 8. So now next, how we can perform the log scale transformation. So let me just paste these two commands in the chat box as well. So the first command, that is dim, it will compute the dimensions of your data, that it will compute how many rows and how many columns are there in your data. The next is summary, it will compute the summary statistics of your data. The next is how we can perform the log scale transformation. So here we are making log, creating an object log. So what we have to do, if you remember last time, whenever we perform a log, we always add 1. So with the df plus 1, and then we are doing the log of, taking the log of this. So then to check whether my data get log transformed or not, what I can do, I can check the summary statistics. So summary of log. So if you look at this now, minimum remains a 0. The maximum here we will obtain 11.429 or 12 point depending on the different samples. So here we will get the, from this we able to know, beside we can also check the dimensions. So this summary statistically clearly let us know whether our data get transformed or not. Next, how we can check the box plot. Now let's draw the simple box plot. So this box plot that we have obtained with the FPKM value when we are taking so this first box plot that I have obtained, that is from the FPKM data. So if you're looking at this, so this representing the all the samples, then it representing the minimum and the maximum value. So it seems like most of the outliers. So now if you make the box plot, so define to differentiate that. So I'm just using a different color with the call. So the first one, without log scale transformation, I'm giving color red. So then we have drawn the box plot for the log data. Here I'm using color blue. So now if you look at this data now, so this is after the log scale transformation. Here if you look at, here we have minimum and the maximum value, how it varies. So then these are, we have obtained box plot for these particular samples. So this clearly indicated what are the differences after performing the log scale transformation. Is it clear to you? So how many of you able to produce box plot in R? So let me just copy this command. You just need to change the data first. So this was when we are plotting box plot for data that contains FPKM values. So how I have done the log scale transformation. In that data, we have added value plus 1 and then we have taken the log of that. And then we stored the this value into an object that is log. So then we have generated a box plot for this particular data. And you can take summary and visualize. Then we can also, if we take one sample, or one gene. So let me show you. I'm taking gene. So in R, how we can extract a particular row or particular column? So here, what you need to do, if you want to take a row, so in that case, I'm taking second row to check whether this be able to. So we have to write before the before the comma. So if in that case, now let's check whether we able to extract a particular gene or not. Sorry. So we have to actually take a square bracket. So if you look at this here, we able to obtain a that is column. So if we want to take a row, okay. So now I can draw bar plot for this gene. Log. Why there more than one similar graph as output files? So it will. So usually it will the script is this running. So if the second, only the last plot that you will get, that is you will get the 2 copies of that. Other graph, the previous one, always you get the one. Yes, both are the same. So last 2 plots are the same. So how many of you able to produce the box plot? Then you can, I think I have to sometimes start just console is. So how many of you able to log transform this data and then able to read your file and then able to produce the box plot for both? Then you can also take a single gene or a sample, then draw a box plot, bar plot for that. So is it clear to everyone? So you need to extract your gene. So how you can extract your gene or a sample? So you can take gene with this simple command. So before the this is the column that you are extracting. So if you're taking before the comma, that is that you are taking a row. So as in R, so this is how you can then plot various kind of plots. So I think you can play with it yourself, and then you can do these exercises yourself, and now we should proceed for the further presentation. So how many of you able to load your table, gene expression table, and then able to log transformation and able to produce at least box plot? The rest of the exercises we can do next time even, and you can do, explore those yourself in the next. So before going into the details of the exploration of the data, let's understand what are different types of data. So data can be vary by the type and the dimensionality. So the first, based on the categorical data. So can anybody give me an example of any categorical data? So editor, yes, gender. It could be gender, location. It could be a kind of tumor types. Like right now we were talking about ER positive, triple negative cancer subtypes, right? Similarly, data can be quantitative. So here we could have a discrete and continuous. Then based on the dimensionality, that is how many features are there. So it could be univariate. If we are talking about a single feature or a single characteristics or a single gene, then it could be a univariate. Then if we have multiple two features at least, then we could have bivariate. So if we looking at gender, it could be male or the female. So that is Y variant. So like for the gene expression data table, when we have multiple variables, that is more than one variables or more than two features, that is multivariate. So now let's understand the importance of statistical analysis of our data. Let's take a simple example. We have two groups. One, we have a males, group 1. In the group 2, we have females. So how we can differentiate these groups statistically? We can consider different measures like its height, its weight, their weight. Then it could be their eating habits like veg and the non-veg. So the simplest test to differentiate these two groups could be a t-statistics or t-test. So in this case, we have two independent groups of the samples, and unpaired t-test can be used to determine whether the means are different for each gene if we are considering like height or a weight. So instead of this example, when we have gene expression data, so what are the important factors that make it too difficult to identify differentially expressed genes? So what are the main considerations? There are thousands of the genes in each sample. Then samples in each group can have significant variation in the expression level. Even when there is a difference we observe, it is hard to know what will be the significant enough difference to say that these are truly differentially expressed genes. So to overcome these challenges, we have to turn various statistical analysis tests. So let's look at one of the such statistical tests, that is t-test. So what are important points to consider to apply a t-test on any data? The first, we can apply t-test only on our data which follow normal distribution, like this well-shaped curve. Then the next, when we are actually apply t-test, next we have to consider the p-value, which represent the level of the significance. So what are these p-value? They represent the probability of obtaining result at least as extreme or a random or a by chance. Since it is not by chance, we can, with what confidence that we can say, for in statistics, p-value less than 0.05 is considered as significant p-value. The next is fold chains. What it represent? So it represents the ratio between the two quantities or the two conditions. For example, we have a condition A and the condition B. So the ratio of the expression of a particular gene in condition 1 in comparison to the condition B, that is what we considered as the fold chains. So now let's look at the formula of t-test, but it represents. So here N1 and N2 represent the size of the group, like how many samples are there in one group, like ER positive or the triple negative, how many samples are there. Then what this S represent, that it represents the common variance of the two groups. So if the absolute value of the t-test is less than 5%, then the difference is considered as statistically significant. Otherwise, it is not. So now let's see how we can apply a t-test in Excel. So we have, when we run this quantile normalization on our data, so we can know our data could be a more normally distributed. So we are going to take that particular data where we have applied and performed quantile normalization. So I think every one of you able to paste that quantile normalization file in the Excel. So let's now see how we can apply t-test in Excel. So this is our data which is already normalized. So now we can apply t-test on this data, because t-test required your data must be normally distributed. So it is considered that after performing quantile normalization, your data becomes more normally distributed. So here we have two groups of the samples. One is ER positive. So I'm just highlighting this group with the yellow, and this is triple negative group. I'm highlighting this with the green color. So if you here we going to apply a t-test. So t-test will compute the p-value. So I'm just writing at the top of the p-value in the header. So how we can apply t-test? Okay. How we can, I can change the color. I can change color from here only directly. Okay. So I am taking ER positive groups. So if we look at this, here we have ER positive group. So I'm giving this color yellow to this, and I'm giving this triple negative sample as the green, so that I am understand what are two different groups that I have. I'm just to differentiate these two groups to understand in the table. So I'm just I have just color them with the different color. So how we can apply t-test? So if you write just t-test, this formula will come. So what it represents, if you click on that formula, it will give you array 1, comma, array 2, then tails and type. So what it represents, array 1 means you have to define which is your first group, right? So if I'm looking at, I have a group from B2 to H2 is my first group. So this is my first array, that is first group. Then next is what is my second array, means the second group. So from I2 till U2. So this is my second group. The next is tail. So tail is representing whether you want to, if one tail or the two tail. If you are familiar with this one tail, you just have to tell that this particular gene is significantly differentially expressed or not. Simple. So two tails means you have to define whether this gene is upregulated or downregulated. So that means we are applying 2. Then type. It is asking what if you go through this t-test formula. So what you will find, there are 3 different types. So the type 3 represent there is variance in each of these group. So that's why we are considering variance 3. So I'm just, if you look at here, what I have done, I have selected first array, then I have defined the second array, that is second group. Then I have just defined that I want to take 2 tail test, and then I have just defined I want to take type 3 test. Type 3 means it represents there is variance in both the groups. So this is what the formula. Once we able to do this, and just click on this. So on the side, if I click on the side, so now I can drag it and apply t-test on all the genes that I have after. So I can remove the extra rows. So now next what we can do, so let me just even copy the gene ID here. So now let's apply filter at all the columns. So next, as we know that we can consider only those genes as significant which have p-value less than 0.05. So we can sort these genes based on smallest to largest. So now you can filter out which of the genes are actually significant in both the conditions. So you can directly filter it out here. So you can select that which genes you need it. So I can, if we are looking at just the p-value, so till here, so 388 genes we have which have significant p-value. So we can filter out those genes. Then the next criteria in the t-test, what was that? That is log fold chains. That is log fold chains. So how we can compute fold chains? So it consider mean of, that is average. So we have to take average. That is B2 to till H2. So this is my group 1. So then I have to define divide the average of that is my second group. So second group, that is I2 tilde U2. So this will compute the fold chain. Then if I just want to compute the log, so I can take log of this value. So this is how we can compute both log fold chain. So what I have done, I have taken the average of group 1 and then divided with the average of the group 2. Then I have taken the log of this part and it is log fold chains. Is it clear to everyone? So this is, then you can consider also the fold chains, how much fold chains that you are interested with, whether you want to take more than one fold chains. So then you can filter out, once you able to filter out genes based on the p-value, only significant p-value, then you can also filter out your genes based on the fold chains, log fold chains. So this is how we can done in the t-test. So now let's move further. So this limitation of the t-test, what is the limitation? That it can be applied only when we have normalized data. When we have, so Aditya has question, what log filter can be utilized? It is plus 1, plus and minus 1 both. So that's the minimum. That if you want to use maximum filter, if you have very large number of genes still and you want to only filter out few genes, you can chase higher log fold chains. So as I have told you that t-test can be applied only t-test or Wilcoxon t-test or Welch t-test, which are actually used standardized t-tests that are applied on when you have normalized gene expression values, like you have already normalized, first you have FPKM values, then you have applied normalized the data using quantile normalization and G-score normalization. So, but when you have count values, so t-test cannot be applied when we have non-normalized value like the count values. So in that scenario, what we can do, so that is there are different packages are available in the R when you have raw count values. So like HR, HTSeq, HTSeq2, these are the different R packages that are specifically designed to apply when you have raw count values. So next we will demonstrate to you how we can apply HTSeq. So what exactly this HTSeq do? That it takes a table of read count as the basis of differential expression calls, and that is non-normalized count of the reads. Then it uses negative binomial distribution to estimate variance and the mean dependence from the read counts. Eventually, it will compute p-value or the and p-adjusted value as well as log fold chains. So now let's go over T-Barn4 platform and see how we can perform differential gene expression analysis at T-Barn4 platform. So this is today I'm going to run only a demo pipeline. So if you have read count values under the utilities section, here we have also. Now let's first go over the platform directly. So if you have a gene expression table at the read count, you can directly run read count under the differential expression analysis. But now I'm going to run only the demo pipeline. So I'm taking the same demo, that is demo RNA-seq. Last time we skipped that step, that is HTSeq. So last time we skipped the differential expression analysis. Now let's run this demo pipeline. So once you are there, just select on, as we are going to run demo pipeline only. Here we have this third project, that is PDX project. We are using the same project here. So the next with the start, be able to upload our files. Then the pre-processing, where we are going to remove adapters first, that is with the Trimomatic. Then next with the PCR clean, we are going to remove PCR duplicates. Next, we are using transcripts, but T, 2T. And then under the RSEM expression table, we have actually selected all the count values, and we can also generate count values with the high set. Then we are going to this select HTSeq2 this time, that is to perform the differential gene expression analysis. Once we are able to do that, then we are going to end this pipeline. We are running the same pipeline, only here we are considering this HTSeq2 part. So here under the download, the folder that here we have, that is HTSeq2 all files if. So if we download this folder, so here the first table that I'm going to show you. what we will obtain. So I'm pasting this in Excel. So here what you've obtained by running this pipeline. So here we have log fold chains. I'm just highlighting it. And then we have the important this p value, then p adjusted value. So what is the difference? Anybody familiar with what is the difference between p value and p adjusted value? So anyone familiar with or know the difference between p value and p adjusted value? Yeah, exactly. It's like FTR. So when we are comparing only two samples, so in that case, we can consider p value as we have multiple testing, multiple samples. In that case, we usually in literature even the p adjusted value is actually considered. So yes, it is adjusting for the different samples when we have a multiple samples. In that case, usually if you have large number of samples, so for the multiple testing, we usually consider instead of the p value, p adjusted value is considered. This is what the log fold chain. This is what first output that we have obtained after running the differential DSEC pipeline. So what are the other outputs? Let's look at them. So then we have also get the output like what are the significant go terms and the pathways. So if you run DSEC2, you will also find out based on the fold chains and significant genes. So what are the different go terms that were found to be significant based on your cutoff that you have selected when you're running DSEC pipeline. So then we have here you will get the different go terms that is associated with the significant differentially expressed genes. So in case you are not obtaining any go terms in the pathway, it means there are not a very large number of significant genes. So in that case, you might not obtain this go terms and the pathways. If there is insufficient number of the genes that are found to be significant differentially expressed. Then similarly, we have this KEGG pathway. This is after running the DSEC2 pipelines. So then similarly, what are different KEGG pathways that were found to be significant differentially expressed in your conditions, in your data. Then we have another output. This is MA plots. So it represents how much fold chains after adjusting the fold chains, how many genes are there. So if you have sufficient number of the genes, then you can also obtain the volcano plots that represent what are the genes after a particular threshold. So these are the major outputs that you will obtain after running the DSEC pipeline. So you can directly give your count values if you have already tables. So then you can run DSEC pipeline directly using this utility option. Under the utility here you have differential expression. You have both the options are here, that is DSEC2 as well as that is HR. So you must understand when you can apply t-test or Wilcoxon or Welch t-test, or when you need to apply these HTSeq or the HR. The major difference, when you have raw read cons, in that case you need to apply HTSeq or HR. If you have already normalized theta FPKM value, then that can be converted into the quantile normalized. Then you can apply any Wilcoxon or Welch t-test. So is it clear to everyone? So type yes. Okay, add it to I. So yeah, as you will, these things will be clear as you can run these pipeline yourself and exactly what outputs that you are obtaining. The basic concept you must understand when you need to apply any kind of t-test or when you need to apply these specific tools like HTSeq and the HR. So let me just share again, go back to the presentation. So t-test and this HTSeq can be applied when we have two conditions. So what we will do, if we have multiple conditions, like here even in our data, if you look at the gene expression table, here we have cancer subtypes, that is ER positive and triple negative. Apart from that, we have mice model, that is a thymic and the nod. So if we want to filter out only the genes that are actually important for cancer subtypes, but not the mice subtype. So the one of the tasks that we can apply, that is the next statistical method that can be applicable in such scenario, that is regression or factor regression analysis. As today we don't have much time, so we will perform the factor regression analysis in our next session, when we are talking about beside we will in the next session we will talk about various machine learning approaches. So regression can be applied, or the factor regression, when we have two independent factors as well as the dependent variables, when we have multiple factors to consider, like here. So if a dependent variable is depend on independent variable by a coefficient and the constant, then regression can be applicable. So we will discuss factor regression analysis as well as the machine learning in our next session. So if you have any questions for today's session, so you can ask. So Editor has a question. What once the samples are large, t-test can be done even if a normality is breached. So usually t-test we usually don't apply t-test specifically pair t-test. So Wilcoxon and Welch t-test only applied when your data is actually normalized. First of all, to applying this t-test, we need to make our data normalized. Is it clear to you, Editor? So if anyone else have any questions? So binomial distribution mark, this is that is negative binomial distribution normalization is applied by HTSeq itself, especially for the read cons. So whatever we have discussed today, so it is explained in detail in the transcriptomics too. So some of the things still we missed, and we can learn that in the next session and go through the transcriptomics too if you want to understand these things in detail. Then for the next session, before the next session, I'm just giving you these assignments. And first, complete Transcriptomics 2 course, and then try to run quantile normalization and HTSeq. You can skip the factor regression analysis as we are unable to run today. And then also try to complete yourself these exercises on OmicsLogic platform in the R, that is data visualization. So in the next session, we are going to talk about data mining and machine learning techniques that we can apply once we have this gene expression data tape. So thank you everyone for joining today's session. If you have any questions and if you face any issue, you can post your query on you can query on the forum as well. Which type of normalization is recommended if I have more than one run file? Exactly, Mark, can you explain run file? Okay, so if you are actually dealing with the same, you have data from the same study or the same experiments, so then we can usually apply quantile normalization if we have normalized already FPKM value or RPKM value. So then we can apply this quantile normalization. So quantile normalization usually considered universal platform independent normalization technique. So still many of the studies suggested that, recommended that, but then depending upon the data, sometimes even the G-score normalization is also useful. Okay. So thank you everyone for joining today's session.
Thank you, Dr. Harpreet, and thank you everyone else. So I just wanted to quickly share my screen and show everyone again how you could make the code OmicsLogic completely free for you, as I just learned that a few people are having some troubles in making the code platform completely free. So just let me know. I will be showing this very quickly and not take up much of your time. So once you're in Code OmicsLogic, you have to go to pricing. And I had sent you a free code for the starter. So if you click on buy now, you will be taken to this page, and you have to just add the promotional code launch 2020. And this would be just 0, and you would just have to check out with your card details and it wouldn't be charged. So this will give you access to the full premium courses. And another question which we are receiving is for learning from the platform. As a lot of you have already taken some of the courses on Edu platform, you may not be able to access all of the transcriptomics courses. So we are going to be shifting to this side very soon, and we are building up the courses up here. So if you go to Learn OmicsLogic, we have those same courses right up here as well. So you could start looking at transcriptomics here as and explore this platform. If any of the courses are not available on Edu, and also you can let me know in case you would want to reset your progress and also take the courses on Edu again. I just wanted to say that it would reset your progress, like your complete progress, and you would have to start over with the courses again. And in case you're not willing to do that, explore them on this platform. So thank you everyone. And if you have any questions, please continue to post them on the forum. And we will be meeting everyone on Wednesday again at the same time. Have a great day, everyone.